Good afternoon, children. So in the previous class, we discussed about autotrophic and heterotrophic mode of nutrition and examples. So in this class, we studied about the detailed phenomenon of heterotrophic mode of nutrition in different examples. So the examples are here, amoeba and paramecium. So in our syllabus, so the nutrition in amoeba and nutrition in paramecium is present. Okay, so actually the amoeba is in a single-celled organism. So especially amoeba, so it mainly made up with the protoplasm without cell wall. Okay, so that's why it doesn't have any particular shape. Okay, so the amoeba food may be taken by the entire surface of the body. So because of it is in a simplest organism, so it has no specific part to take the food material, so they take food by the entire surface. So but coming to the multicellular organisms, so the complexity is increased under the different organs, and the cells are formed the different tissues, and tissues are formed the different organs, and the organs are formed the different parts. Okay, the parts become specialized to perform the different function. So which means, so a part which is used for the obtaining of the food material, and some parts which is used for the digestion of the food material, and some parts which is used for the excretion of the food material, waste material, and etc. Okay, so just you see that pic how so how amoeba involves in the digestion process. Okay, so see this video. So it is an animated video of amoeba nutrition. Okay, amoeba is in a unicellular single cellular organism. Okay, so actually the food is vary, already I told you in the discuss every organism and every cell, so that food taking is vary from one cell to the another cell. So it is in a nucleus, and this is in a protoplasm and covered with an endoplasma membrane. So these protoplasm that produce finger like projections, so which are called as these finger like projections, which are called as pseudopodia. Okay, so this pseudopodia goes towards the food material and engulf the food material. So immediately what will be formed? A food vacuole is formed here. Okay, after the formation of the food vacuole, so these food particles are entered into the protoplasm. These food particles are entered into the protoplasm and digested by the different hydrolytic and proteolytic enzymes. So after the digestion, what will be happen? So the food particles are diffuses throughout the protoplasm. So whatever the undigested material that present in the protoplasm is expelled out from the body. Okay, so this is a nutrition in amoeba. Okay, so here the engulfing of the food material by the entire surface, okay, that nothing but it is called as endocytosis. What it is called? Endocytosis. Okay, so this you see this microscopic real video of the amoeba. So it is an amoeba that produces the pseudopodia. So these are the finger like projections, and it is in a food material. This unicellular organism is in a food material automatically. So here both the pseudopodia are connected to form a food vacuole here. So after the formation of the food vacuole, the organism is died. So here they release the enzymes, and those enzymes are digest that organisms, and after that the digested material is diffused throughout the protoplasm of the amoeba. So it is called as nutrition in amoeba. So just you see here, so the cytoplasm release the enzymes and the cell is digested. Okay, so this is called nutrition in amoeba. So coming to this next example, so that is in a paramecium, nutrition in paramecium. Okay, so paramecium is also called as a unicellular organism. So it is also called as slipper mold. So if you see that the parts especially present in the paramecium, so it has a groove like structure. Okay, so these groove like structures, what it is called, so it is called as cytostome. What it is called? Cytostome. Okay, so with the help of this cytostome, so it take the food material, so there is a small difference in between amoeba and paramecium. So amoeba take the food material from entire surface of the body, but the paramecium is, it take the food material at a with a specific part. Okay, so this specific part is nothing but it is called as oral groove or cytostome. So with the help of cytostome, so it obtain the food material, and that food material is entered into the buccal cavity. Okay, so after that this food material is formed a food vacuole, and this food vacuole is travel to the cytoplasm. So in the cytoplasm, what will be happen? The enzymatic digestion is takes place. So whatever the complex molecules that are digested and to form in a simple molecules. So these simple molecules are absorbed by absorption is takes place. That nothing but absorbed by this protoplasm, and whatever the waste material, so that is discarded from the cell. So through a particular pore. Okay, through a particular pore, so nothing but it is called as an anal pore. So it is called as an anal pore. So just you see that here the paramecium nutrition video. So these are the paramecium cells. It is in a real video of the paramecium. Okay, so now you see that yeah, how to paramecium eat the yeast cells. Okay, yeast is also one unicellular organism. So actually the yeast cells that are mainly stained with the congo red stainer. So this you see, so it is an outside of the yeast cell is present here. Okay, so the yeast cell outside, so automatically so already it is taking some yeast cells from external body. Just you see, watch this video clearly. So just you see, so from this pot only, so it will take, so this pot is nothing but what it is called, cytostome. The pot is called as cytostome. So from here only, just you see here. Okay, so here, so they take the food, nothing but it is an yeast cell, and the yeast cell, when it is enter into that one, automatically the cytoplasm of that protoplasm of the paramecium is also shows Congo red color. So because of before that, so we stained that yeast cell with a Congo red. So it is a real microscopic nutrition in amoeba. So nutrition in paramecium videography. Okay, so these are the two examples for the heterotrophic nutrition in amoeba and as well as in paramecium. Okay, so now coming to that next slide. Okay, parasitic nutrition in cuscuta. So actually the cuscuta is in a leafless twining parasitic plants, so belonging to the morning glory family, so which is also called as a convolvulaceae family. So it is in a leafless twinning. Okay, so the twinning nothing but it is in a climber. Okay, so it is in a parasitic plant. So because of it is in a non-chlorophyllous plant, okay, that's why it is also called as parasitic. Okay, so the genus cuscuta nearby 170 twining species, so they are distributed in the temperate and tropical region of the world. So especially here the dodder contain no chlorophyll. So due to the loss of chlorophyll only, so they are not prepare the food material. Okay, so coming to that cuscuta reflexa, so it has a small amount of the chlorophyll molecule. Okay, so here the cuscuta produced a root like structures which are called as hastoria. So these hastorias are penetrated into the tissue of host plant, and these hastorias are helpful for the absorbing of food from the host plant. So this is a main phenomenon that involves in the absorption of food from the host plants by the dodder plants. Okay, so you see that this picture, so it is in a yellow color twined. Okay, yellow color twined cuscuta plant is present. So if you see that these are the flowers. Okay, so actually the slender string like stem, so that is mainly yellow in color. So as in some plants it also shows orange, pink or brown in color. Okay, so the leaves are reduced to minute scales, so you do not observe any leaf. So whatever these green color leaves that are having only the host plant leaves. Okay, so actually the twine, so do not have any leaf. Okay, that's why they are reduced into the scaly leaves. So here this you see that flowers. So these flowers are nodule like clusters. Nodule like clusters, and many flowers are arranged in a node, that nothing but it is called as a cluster. Okay, so which is yellow or white bell like petals. So if you see that petal, it is in a yellow or white bell like petals. Okay, coming to the next slide. So here the dodal tree. Okay, so the dodal plants. So how the seeds are germinated. Okay, so the dodal seeds are germinated forming a anchoring root. So first they form the anchoring root, and they produce the slender branch, slender stem or slender branch that grows towards the host plant in a spiral fashion. So after that, what will be happen? The seed is germinated and it produces a slender stem, so that grows into a spiral manner until it attaches to the host plant. So when it is attached to the host plants, automatically, so the slender stem, so which connect with the root system, is disintegrated. So now the stem, so which is attached to the host plants, is produced a finger like projections which are called as hastoria, and these hostorias are penetrated up to the xylem and phloem of the tissue of the host plant. Okay, so when these hostorias are produced into the xylem and phloem tissue of the host plant, from the xylem they absorb the water, and from the phloem they absorb the food material. Okay, so here just you see it, the anchor root and disintegrate with the host stem, then it is grow and obtain fruit from the host plant with the help of haustoria. So finally it is developed as a web-like structures on the host plant. So if you see this, okay, so now the dodder plant is germinated. Okay, so when it is germinated, automatically it produces a slender stem. Okay, so this slender stem is attached to the host plant. Okay, these dodder seeds are germinating now. So you watch it. Okay, how beautiful it is. Okay, now so it is in a host plant stem. It is in a host plant stem. Okay, so now it is in a slender stem. So it surrounds the host plant stem in a spiral manner. So just you see this stem of the dodder is surrounds the host stem. Okay, so just you see, so now it produces the hastoria into the host plant. So just you see here, it is bulged, the stem is bulged, so it produces in a finger like projections that are inserted into or penetrated into the host plant. So after that, what will be happen? The stem connected with the root system is disintegrated. Just you see this part. Okay, so it is disintegrated or rotten. Okay, so this pot is continuously grow. Okay, so this is nothing but it is called as a parasitic nutrition in cuscuta reflexa. Okay, so now coming to the next slide. So it is an overview of the modes of nutrition in plants. So actually the plants that contain the autotrophic nutrition and heterotrophic nutrition. So autotrophs are mainly divided into the photoautotrophs and chemoautotrophs. So especially the plants are that shows the photoautotrophs. So coming to the chemoautotrophs, especially the bacteria which involves in the chemoautotrophic mode of nutrition. Okay, now coming to the heterotrophic nutrition, so already we discussed about the parasitic and saprophytic. Okay, along with these two, parasitic and saprophytic, the symbiotic association is there that we will discuss in the later class. Okay, examples for that lichen. Okay, and also the insectivorous. Okay, insectivorous plants. Okay, these insectivorous plants, the examples for that insectivorous plants are Drosera and Utricularia and Nepenthes and Dionia and etc. Okay, so now coming to the next slide. Okay, the heterotrophic mode of nutrition. So first one is in a holozoic nutrition and saprophytism and parasitism. Okay, the heterotrophic is mainly divided into three types. One is in a holozoic, and second one is in a saprophytic, and third one is in a parasitism. Okay, so here the organism feed by ingesting solid organic matter which is then digested and absorbed into their bodies. So examples, women. So women are feed the different kinds of the solid materials. Okay, after that solid material taking or ingesting, so those solid materials are digested and absorbed by the body. So this type of nutrition is nothing but it is called as a holozoic nutrition. So coming to the saprophyte, I already told you they feed on dead and decaying matter. So especially the bacteria and fungi are digest the food material externally and before the nutrients absorb. So now coming to the parasitism. So to obtain the nutrient from living organisms, either it may be the plants or it may be the animals. Okay, so this is called as a flies, lice, tapeworms. Already we discussed in the previous class. Okay, so these are the questions, you note it. So the first question is tabulate the autotrophic and heterotrophic nutrition with suitable examples. And second question is explain the nutrition in amoeba and paramecium with suitable examples, suitable diagrams. Sorry. Third question is what are the raw material for process of photosynthesis? And fourth question is what are the end products of photosynthesis? And fifth question is if you have a chance to meet a nutritionist, what kind of question do you ask about the heterotrophism? Okay. And next sixth question is how do you support the statement plants are primary producers? Okay, so these are the six questions, you note it and write the answer. So with the refer with the textbook. Thank you.